Hi, good afternoon. My name is Yewon Chun, and I'm a director of the summer internship program and postbac program in the Office of Intramural Training and Education. And today I am so pleased to introduce Dr. Eric Green, our first speaker for the summer lecture series. Dr. Eric Green is the director of the National Human Genome Research Institute, NHGRI, at the National Institutes of Health. Since 2009, he came to NIH as a principal investigator in 1994 and served as a director of the NIH Intramural Sequencing Center, chief of the NHGRI Genome Technology Branch, and later became the NHGRI scientific director. Dr. Green received his BS degree from the University of Wisconsin-Madison and his MD PhD degrees from Washington University in St. Louis. During residency training, he started his career in genomics research. Dr. Green's research was at the forefront of efforts to map, sequence, and understand eukaryotic genomes. His work included significant involvement in Human Genome Project. He has authored and co-authored over 340 publications. That's a lot, right? Today, Dr. Green will talk about his research and how genomic knowledge is being used by researchers and clinicians to tackle increasingly complex problems in biomedicine. Please join me in welcoming Dr. Eric Green.
Well, thank you very much. It's a pleasure to be here. I'm always delighted to speak to the summer interns and summer trainees. I do it every year for the summer trainees who are at my institute, NHGRI, and when I was invited to do this for the entire NIH program, I immediately accepted and was looking forward to spending this afternoon with you. As you can see from the title, and you may have gotten a little hint of this from the introduction, I'm going to talk about this field of genomics that I've been involved with actually since the inception of the field. But I'm really going to describe for you a number of journeys, which is why I have the word journey specifically in the title of this talk. It's going to be a journey about this field of genomics and where it's come from its very beginning. It's going to be a description of this new emerging journey for precision medicine, which I'll talk about near the end of my talk. But I'm also going to interweave with those two journeys my own personal journey, because one of the things I was asked to talk about to an audience like this is a little bit about my own story, my own journey, since all of you are about to enter your professional journeys. And sometimes it's illustrative to hear what others have done before you. And so I thought you might be interested to hear some things about really very much how my own personal journey has been interwoven with the journey of genomics and increasingly the journey of precision medicine. Now, as you heard in those introductory comments, I was born and raised in St. Louis, Missouri. I went to the University of Wisconsin as an undergraduate, and then I came back to Washington University having made the decision to pursue an MD PhD degree. That was a key decision. I wanted joint training to be both a physician and a scientist. Here I am at my medical school graduation ceremony. I show this picture not so you could see me as a youngster, but rather I want you to note the date because it's a relevant part of this journey. I got my MD PhD degree in 1987. Hold that thought. We'll come back to it in a bit. I then had another decision to make. What was I going to do? Was I going to do a research career? Was I going to become a physician? Was I going to try to do both, do some research and do some clinical work? And that was another key decision I made. What I gravitated to was an area of pathology known as laboratory medicine, sometimes referred to as clinical pathology, which is the part of pathology that really runs the diagnostic laboratories in hospitals, the blood bank, chemistry lab, microbiology lab. And what I was sort of vaguely interested in was the idea of DNA diagnostics, as illustrated in the bottom right of this slide, thinking that there were some exciting developments going on in molecular biology and the use of DNA information, possibly for medicine, and maybe this would be a nice way to bridge those interests and be involved clinically through laboratory medicine, but also provide me time to be a research scientist as well. And one of the other attractive things about a residency in pathology, whether any type of pathology residency, is it gives you time to get back to the research lab to pursue postdoctoral research. And so after my first clinical year in laboratory medicine, I was allowed to pick a research laboratory to pursue postdoctoral training. And I was very definitive in what I wanted to do at that point when I realized that I could work in a laboratory that was just starting to work on problems related to this brand new field of genomics. Now, 1987 comes up again here for a very deliberative reason. The field of genomics did not exist before 1987. In fact, it didn't even exist by name until 1987. In fact, the first use of the word genomics in the scientific or medical literature happened in this inaugural issue of a brand new journal called Genomics, where the editors basically wrote an editorial and talked about this new discipline, a new name, and a new journal, and for this newly developing discipline of genome mapping and sequencing for which they adopted the term genomics. So I just make this point that I got attracted to this field basically at its inception. And it was in 1987 that this field first got named, and yet I graduated in 1987, which means never once during my formal education, medical school or graduate school, did I ever hear the word genomics once because the word didn't exist. And yet here I am leading the institute at the NIH responsible for genomics research in the United States and really the leader in the world for genomics research based on the activities of what my institute is responsible for. Now, what is genomics and why is it that 1987 was a year for starting a new scientific discipline? Well, let me just take you back to some basics. I actually suspect all of you probably know fundamentally what genomics is, but a little bit of context probably would be helpful to bring everybody along. Think about the concept that the human body has something on the order of 10 to 20 trillion cells that make up the human body. And I could tell you that for decades, if not centuries, I think scientists and physicians were intrigued by this idea that all those different cells in our body, many of which operate in very different ways, and yet they all operate off the same fundamental blueprint. How is it that all those trillions of cells, whether they're operating in the brain or the muscle or the liver, all have the same fundamental blueprint? And wouldn't it be intriguing to understand that blueprint? Well, as time marched on, we came to learn that that blueprint was housed in the nucleus of cells and these structures known as chromosomes. But while hints came along in the early 1900s, it really crystallized our understanding of that blueprint molecule in the 1950s when Watson and Crick made that discovery about the double helical structure of DNA. It was the last piece of the puzzle that gave us insights about how it was that DNA was the blueprint, in other words, the information molecule for heredity. And what I saw basically transform the field in the 1970s and 1980s was better and better and better methods for studying DNA. And in particular, when I was a graduate student, certainly that was the time of the molecular biology revolution, when all these techniques came about for basically cloning, isolating, manipulating DNA in a laboratory. And the late 1970s were when those first methods came available for actually reading out the building blocks of DNA, and of course, the ability to read out the G's, A's, T's, and C's, the order of which, of course, encodes all the information necessary for life. That is the blueprint information, the order of the G's, A's, T's, and C's. Well, the notion by the late 1980s was that, wow, we've gotten really good at isolating and manipulating and reading DNA. Maybe we could actually think about reading out our entire blueprint. And if we could get really good at sequencing DNA and do it in an organized way, maybe we could read all the DNA of humans and other organisms. And all the DNA of a human or an organism is its genome. And that led basically to the creation of this field of genomics, the comprehensive study of the DNA of an organism. And what became particularly attractive was this idea that, while humans had a lot of DNA in its genome, it's not infinite. It's about 3 billion letters. It's 3 billion G's, A's, T's, and C's make up the human genome. And would it be possible to be able to get organized enough to read out all 3 billion of those letters once and for all and make that fundamental information about the human blueprint available to all scientists around the world? This was conceptualized in the late 1980s and organized into a project that launched on October 1 of 1990, something called the Human Genome Project. Once again, I was very fortunate. I was in the right place at the right time. I was at a place that was doing some early genomics work, and I had the ability to basically join the Human Genome Project as a postdoctoral fellow on day one, on the front line, and participated in the project from beginning to end. I was a resident at the time. I was a postdoctoral trainee, but I was able to amalgamate all these things and basically be involved in the Genome Project really at its complete inception. The Genome Project was incredibly successful by almost any metric. 13 years later, the project was completed. We accomplished all of our goals. We delivered the first sequence of the human genome. And to some extent, the rest was history. It was 15 years and several months ago when the Genome Project concluded. And it was truly a euphoric time for biomedical research because there was plenty of skepticism about whether the Genome Project would be successful at the beginning. But having it be so wildly successful was sort of a validation of the ideas of why we wanted to pursue sequencing the human genome in the first place. I'll come back to the storyline of the journey of genomics, but let me just say a couple more things about my own storyline that I often get asked, especially as a director of an NIH institute. And sort of like, well, what was sort of the different stages of your career, and why did you make these decisions, and how did you ever become an NIH institute director, and so forth? And I will tell you that I have not been the same over the roughly 30 years of my professional career. I've certainly had different personas. And I sometimes sort of joke about how different I have been in different points in my career. So for example, I got involved in the Genome Project from the very beginning. I ended up having my own laboratory where we worked on the Genome Project and worked on other things. As you heard, 24 years ago, I got recruited here, and I had a very active research program here for a number of years. I had plenty of summer students come work on the Genome Project and then activities after the Genome Project. And I would say, if you sort of ask me, well, what's a good persona that describes what you were like during the Human Genome Project, what you were like when you were running your own lab, I would think about some movie characters, and I would say the persona that matches me perfectly almost is this guy. It's Emmett Brown of Back to the Future. I mean, very energetic, crazy ideas, going a million different directions, talking very fast. Yeah, that was me, especially during the Genome Project and a few years beyond. But then opportunities come up. You heard during the introduction, I had opportunities to take on leadership responsibilities. I eventually became the director of our intramural program. And then about 8+ years ago, I had the opportunity to become director of the institute. And now I don't get to have as much fun. Emmett Brown always had fun. Now I have a whole different job as directing an institute. Now if you said, who's your persona? What are you like on a day-to-day basis in a job as an institute director? I would say, really, it's more like this. I'm this guy. That's just sort of like, I don't really do anything. I just wait around. People bring me issues and problems, and I go in my booth, and I turn some dials, and I send some emails, and I try to solve some problems. It's much different. It's very gratifying, to be honest with you, but it's much more like the Wizard of Oz, and it's like Emmett Brown. Now, often I'm asked when I talk to undergraduates, they say, well, you were really smart. You had this great plan about how you were going to work on the Genome Project, and then you were going to go to NIH, and you're going to eventually be an institute director. How did you ever formulate your life plan? And I would say, you must be kidding me. I never had a plan whatsoever. I had no idea what I was going to be doing when I was your age. I had no idea what I was going to be doing at multiple stages in my career. My life, all of your lives, this is what it's all about. You're going along, and then you come to a fork in the road. You either go right or left. That's what it comes down to. You decide, do you want to go to medical school? Do you want to do graduate school? You want to do neither? You just go one way or the other. And then a year or two or 3 or 4 or 5 later, you come to another fork in the road, you make your decision. I could tell you about 8 different times I came to a major fork in the road, and I just had to go one way or the other. I never really had a grand plan that I was going to take 3 rights and then 2 lefts. It just doesn't exist. I got to tell you, I firmly believe that, yeah, you can have a general plan what you want to do in life, but then a lot of your life is about improvising. It's about just making a decision whether you're going to go right or left. And the truth of the matter, life is just that small interface between the two. And I could point to multiple times in my life where I just had to improvise. And I made decisions. I don't know if they were the best decisions, but I've ended up in this position, and I've had a very gratifying career. But trust me, when I was your age, I was at your stage, I had no idea what I was going to be doing 2, 3, 4 decades later. So that's enough about my journey. Let me return to the journey of genomics. So where I left off in the journey of genomics is that we had completed the Human Genome Project. So now I want to tell you about sort of some of what I think are the key advances, the real highlights that we've seen in the first 3-ish decades of genomics. We've gone from 1987 to now 2018, 31 years of genomics. What have been the big highlights? I'll tell you about 6 of them because I think they nicely showcase what we've accomplished in this young discipline. I actually already told you about the first one, and hurrah, the first highlight will always be the first highlight. We sequenced the human genome for the very first time by the Human Genome Project. We laid out the order of the 3 billion letters that make up a reference human genome sequence. And what I could tell you is that having completed that 15 years ago, oh my gosh, has genomics caught fire and spread across the biomedical research landscape. Actually, it's spread even beyond biomedical research. The applications of genomics have been breathtaking, and they have been wide and far. Shown here is a subset of the areas that have been profoundly influenced by genomics over the past 15 years. Each one of these could be a lecture in and of themselves. I'm actually not going to touch on really any of these 9 areas. Rather, the area that I'm going to emphasize is the area that I work in. I'm going to emphasize the implementation of genomics for studying human health and disease and possibly for changing the practice of medicine. And of course, the reason I'm going to take this more medically oriented track in describing accomplishments in genomics is because where we are here. We're at the National Institutes of Health, all about turning discovery into health. And in particular, the institute that I've now been at for 24 years and have now led for 8+ years, the National Human Genome Research Institute, we were originally created by the US Congress to lead the US's effort in the Human Genome Project. But, well, 15 years ago we finished that part of it, so we had to expand what we were going to do. And now we think about we really want to advance human health through genomics research. So then the question becomes, okay, let's think about it. 15 years ago, wow, all right, you have a first sequence of the human genome. How are you going to go from that to actually changing the practice of medicine through a better understanding of the role of genomics in health and disease? And what I would tell you is we are progressively moving in that direction. And in particular, what I have emphasized and really have helped push the Institute over the last 8, and really the field, over the last 8 years, is to foster these medical applications of genomics and to realize this emerging discipline of genomic medicine. Now, by genomic medicine, I mean using genomic information about an individual as part of their clinical care. And here's the key. Each one of us is a little different than anybody else in this room. And yet so many aspects of medicine, we treat everybody sort of like they're the average patient. But we all know each of our blueprints is a little different. If only we can use unique blueprint information about individual patients, maybe we can tailor their medical care and do better than what we're doing now. That is the idea of genomic medicine, using genomic information to guide the care of patients. Now, you'll hear other words sometimes used for this, other synonyms, individualized medicine, personalized medicine. We'll talk about precision medicine later. These are all sort of synonymous, but genomic medicine is very narrow in that it's just talking about genomic information, not other types of information about an individual, just about genomic information, and that's what I will emphasize. Well, that sounds great. Let's do it. Yeah, but how do you do it? How do you progress from the most fundamental information about our blueprint that the Human Genome Project produced to actually changing the practice of medicine? Well, this is going to be a journey. It's certainly going to be a hard journey. It's going to not involve a single step. It's going to involve multiple steps, and we can't possibly imagine we know what all those steps are. But like any other journey, you get started, you figure it out as you go. The other thing about this journey is that it's not going to happen quickly. We know it's not going to be a sprint, and we're not going to do it alone. It's not like it's going to be 2 or 3 scientists and clinicians are going to sort of figure it all out and then just tell all the rest of us. This is going to be much more interdisciplinary. It's going to require all sorts of different people from different backgrounds all working together to basically slowly and very methodically figuring out how we're going to use fundamental information about our genomes to practice medicine in a different way. The analogy I would make is not a sprint, but more like a marathon. And I like the marathon because there's many people running shoulder to shoulder with many different ideas, many different expertise areas, all brought together with a common goal. So that's the vision for this journey. But how do you actually make that happen at a funding agency like here at the NIH or in my institute, the NHGRI? Well, I will tell you that one thing that has benefited us well is to engage the scientific community to help us basically develop guides, develop plans, strategic plans, that will sort of tell us what we might need to accomplish each of those steps that would inch us closer and closer to the realization of genomic medicine. And so we have very much benefited from this. We benefited from it during the Human Genome Project, and ever since then we've benefited as well. We sort of see this progression from base pairs to bedside, or if you prefer the metaphor, from helix to health, as really being a journey that needs to be guided along by expert advice on a continual basis. So along those lines, we engaged the scientific and medical communities for the 2 years leading up to the end of the Human Genome Project. And then the day the Human Genome Project ended, we published in Nature, the Institute published this strategic plan that sort of said in 2003 what needed to be done over the next 5 to 10 years to make genomic medicine a reality. Although we didn't even use the word genomic medicine then, we just sort of said to apply it to human health. And it really served us well, and I'll show you one vivid example of that in a minute. But a good strategic vision has to be updated, because no matter what you think you know now, 5 years from now, 10 years from now, you have a much clearer vision of what's needed. So by 2011, we updated our strategic vision with this strategic plan, published in Nature again. And in particular, notice the word genomic medicine actually appears in the title. We laid out in much more detail what were the steps that were going to be needed to actually see genomic medicine become a reality. So these two guides have basically helped show us the steps that were needed in this journey going from the Human Genome Project to the realization of genomic medicine. Well, with those as the guides, what have been some other highlights? Well, I would certainly tell you that the number 2 highlight, and I think this will be number 2 on this list for a long time, has to relate to the technologies that we have developed over the last 15 years to sequence DNA. And it's been remarkable how quickly and how effectively these technologies have been applied for use in a research laboratory and increasingly in the clinic. And what we have seen happen in 15 years since the end of the Human Genome Project is a precipitous decline in the cost for sequencing a human genome, such that now I can tell you that the cost for sequencing a human genome has been reduced by nearly 1 million-fold. Now, this did not happen by accident. In fact, I want to bring you back to, as I promised I would, the strategic plan that we published the day the Human Genome Project ended in 2003. And we talked about a lot of things that needed to be done, having completed the Human Genome Project. But among the real highlights and the important things that we said is that we said that there were technological leaps that seemed so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. And the example that we gave, which was a pretty audacious example, was the ability to sequence DNA at costs that are lowered by 4 to 5 orders of magnitude than the current cost, allowing a human genome to be sequenced for $1,000 or less. Now, our names went on this paper, and yet we gave as the example the notion of sequencing a human genome for $1,000, a nice rounded number. Why was that so audacious to put our names on a scientific paper making a claim like this? Well, the reason it was a little bit audacious is because this was the day the Human Genome Project ended. We had just sequenced our first human genome ever, and it cost something on the order of $1 billion, roughly. And here we were saying, yeah, we'll just come up with better technologies, and we'll knock 6 zeros off of that figure, and we will deliver what was known as the $1,000 genome, which actually seemed like a very reasonable clinical test. It was the reason why we picked $1,000. It was very rounded, and it was very affordable from a medical point of view. That was about the cost of an MRI or a CAT scan back then. And that was the goal. The only problem is we had no idea how we were actually going to do that. We just said, that's what we need to do. And what was really particularly challenging about it is you have to realize how the human genome sequence was actually generated by the Human Genome Project. It was generated in factories that look like this. This is an actual photograph of one of the main laboratories that sequenced the human genome for the very first time. There were dozens of these laboratories in multiple countries around the world. They were operated by literally thousands of people working in these laboratories. They would operate 7/24, generating data. Those machines, which were the machines that were then used to get the Genome Project across the finish line, were about the size of this podium. It's about the size of them, and you would just constantly feed them to generate data. And here we were, and that's why it cost $1 billion, right? I mean, big equipment, lots of people, production 7/24, and so forth. And here we were saying, well, wow, we just need something new. We need something better. We need something quicker. We need something disposable. We need some fancy schmancy device, such as shown in an iconic form here. And maybe then we could get sequencing to be so cheap that we could sequence a human genome for $1,000 or less. Well, the good news is that we've sort of done it. It didn't happen by accident. Our institute, for example, developed a major technology development program. We gave out lots of grants to lots of people in lots of places, universities, even companies, research institutions. Some ideas were totally wacko, but we wanted them to test them ideas, and some of them were really quite spot on. And a lot of good new ideas and new technologies came out of that. In addition, though, we can't take all the credit. The private sector got quite involved. Multiple companies came up. And it was a beautiful example of both the government-based funding and private sector funding, each doing their thing, helping to feed new intellectual ideas into these companies that get commercialized. And to some extent, the rest is history. It's worked extremely well pretty quickly. Shown here is a story from Nature from a few years ago, talking about how close we were to a $1,000 genome. Shown on the bottom left is a graph that we keep track of. Notice it's logarithmic on the Y scale, and notice in green the precipitous decline in the cost of DNA sequencing, or sequencing a human genome, starting about 2007 when some of the new technologies got introduced, and you can see the cost just go plummeting down and now sort of hovering around the $1,000 mark. But what's really remarkable about it is just these new technologies that just keep popping up, better and newer ones. So shown on the far right, for example, is sort of kind of the state of the art at the moment where a lot of things are done. They're little chips. That's what the photo is. I'm going to show you in my hand. Sort of, you can see it looks something like this is one of those devices. There's, little slides that you put little samples on here, and then these little chip-like devices get put into instruments that actually would fit nicely right on top of this podium. So we've gone to a size of being as big as this podium, now something that would fit very nicely on top of this podium. And wow, you can sequence genomes quite efficiently and quite effectively. But it's not just the cost, it's also how rapidly you could do it. Because that's what's really also quite remarkable. What I told you before was that first human genome sequence cost about $1 billion. But what I didn't tell you is that we were actively sequencing human DNA for about 6 to 8 years to get one human genome sequence. The good news is nowadays, using these fancy chip-like based devices and so forth, it's down to almost $1,000. We'll probably go below $1,000 fairly soon. But importantly, we could generate a genome sequence in about a day or 2 or 3. That starts to become a realistic clinical test. You don't want to wait around 6 to 8 years. You're willing to wait around a day or 2. And so that has been also really remarkable. Now, the other thing that's remarkable is I don't worry about this stuff anymore. I used to worry about sequencing technology. I am quite optimistic on what's happening with what we currently have, but more importantly, what's likely going to come in the coming years. In fact, any of you who are involved in sequencing DNA these days, whatever technology you're using, probably by the time you're a graduate student or certainly as a postdoc or a physician, we'll have all new technologies. There's many, many new platforms that are still being developed and at different stages of commercialization. I thought you might think it was fun to sort of see what the latest one is. So I told you first we started with an instrument as big as this podium. Then we have another instrument that we've talked about that sits on top of the podium. But the real rage going on now, just being tested quite extensively, is something called nanopores, which basically involve little pores that sit in membranes, and you pull strands of DNA through, and you measure different things that allow you to detect exactly which base is being pulled through in real time. But what's remarkable about nanopores is that everything gets miniaturized. And so this is actually one of the devices. It's sort of out of the leading company. This is the entire laboratory in my hand. That's all you need. Basically a lab on a chip. I open it up. There's a little chip in here that comes out. You basically apply your sample. It could even just be blood. You apply to that. That's all you need to do. You don't need a laboratory. You then take this device, which is a USB device, and you plug it into a laptop, and you can sequence DNA. And this is not science fiction. You really can sequence DNA. And you can actually sequence DNA using either a Macintosh or a PC laptop. Pretty impressive, right? And it works well on both. And it's incredibly good for sequencing small genomes, like bacterial genomes, viral genomes. It's getting better and better for sequencing human genomes. Not quite there yet, but getting pretty close. The portability and the expense is just as cheap as could be, and it's incredibly portable. So this exact device, guess what, was used in the field in Africa during the Ebola outbreak several years ago to type Ebola viral strains in different villages. Didn't need a laboratory, just needed a laptop. Similarly, this exact device used up in Antarctica during an expedition to sample different analyze different environmental samples. And this device was used to sequence DNA in space for the very first time on the International Space Station. Pretty cool. All true. And so this is the kind of technology we can expect that will probably flow in the coming, and other technologies will supplant that thereafter. In fact, this same company has claims that they won't even need a laptop because they have basically an early design of a little device that will attach to your iPhone or your smartphone and sequence. And this is not a joke. This is serious. It probably will come to pass. And so this is the kind of excitement we have around technology. And you're going to just see waves and waves of new technology. I think sequencing DNA will get cheaper and cheaper. So I really spent a lot of time talking about this because I really want to make an important point here. And I've seen this, I've read about this, I've seen some of this, and I think you may see this in your career, maybe not in genomics, but in some other area. Technology advances really do drive science. It's why genomics has been so remarkable, especially over the last 15 years, is the technology surges. You go back in history, Wow, when the first telescope was invented, it changed the whole field of astronomy. Wow, when that first microscope was invented, changed the whole field of cell biology. When that first scanning device, the CAT scan-like device, completely changed radiology. Guess what? These sequencing instruments, they're changing genomics. It's changing biomedicine. It's going to change medicine. And it's all because the technology has just driven this so hard and so fast and so remarkably effectively. So that has been the second highlight on my highlight reel. I want to take you to the 2011 strategic plan because it will sort of tell you a little bit about the progression for the next set of highlights. In our 2011 strategic plan, the journey that we described starting in 2011 was one that moved us along from left to right because we mostly had been learning a lot about the structure of genomes on the far left. We realized that having that technology capabilities, we needed to now expand our analyses to better understand the biology of genomes, eventually use that information to understand how genomics plays a role in human disease. And if we could get that far, we'd be able to get insights about advancing the science of medicine, and eventually having ideas for improving the effectiveness of healthcare by using genomic information. This progression really became a guide, if you will, or the kind of steps that were going to be needed to realize genomic medicine. How have we done on seeing those steps come to pass? And I think we've done well, although we certainly have a lot more to do. I would say the third highlight really relates to this idea that the reason we generated better ways of sequencing genomes is because we wanted to sequence genomes. We didn't want to just sequence one genome. We're not interested in a hypothetical reference genome alone. We're interested in our genomes. And we're interested in how our genomes differ from everybody else's genomes. Eventually, we want to be interested in our patients' genomes. And so we want to sequence a lot of people's genomes, and the good news is we've done that now. We've sequenced tens of thousands, actually we believe it's probably hundreds of thousands of human genomes have now been sequenced. And the reason we did that and continue to do that is we want to know not how we're the same, that's the boring part. We want to know how we're different because it's our differences in our genomes that really are incredibly important for our health and well-being. So let me just sort of make sure you appreciate a little bit about human genomic variation. So if you take a stretch of DNA in your genome, for example, any given stretch, and you scan across it, you basically differ about 1 out of 1,000 bases compared to, let's say, the person sitting next to you. So all of us are 99.9% identical, but that means about 1 out of 1,000 places we're different. And you can see the differences. I'll just sort of pop up here to illustrate it. And so your genome, you have about 3 to 5 million differences in your genome, single letter differences compared to the person sitting next to you. You have other kinds of variants, the structural variants and various other kinds. But I'm just talking about, we just keep it simple and look at single letter differences, single base or single nucleotide differences. And you have about 3 to 5 million of them. But by the way, most of those 3 to 5 million variants, somebody else in this room probably has. Most of your variants are common variants. They exist in a few percent in the population. A very small fraction of your variants are relatively rare, but most of them are quite common. And of course, we're interested in that. I mean, we would love to have a deep catalog of the most common variants that exist in the world's populations. Well, why would that be useful? Well, we really want to know which of those variants are medically important or functionally important. Turns out that most of your 3 to 5 million variants are inconsequential. They have no influence on your physiology, your health, or your well-being. But a subset of your variants are consequential. And we'd like to know that. We want to know those subset of variants that maybe aren't the best variants, and maybe they confer a risk for getting a disease. But then there's other variants that you want to know about because they actually may protect you from getting a disease or be associated with some other positive attribute. And the idea was, since most variants are relatively common, wouldn't it make sense to go generate really big catalogs of the most common variants that exist in the world? Put them up on the internet for everybody to study, and then have scientists go through and try to figure out which variants are important, which ones are not, which ones influence the genome and how it functions, which ones are not, and do this very systematically. And that might be a path to figuring out which of the variants can we ignore, which of the variants do we want to pay attention to. And so there have been a number of projects that have been performed since the end of the Genome Project that have basically tried to sequence lots of people's genomes, catalog the variants, make them available on the internet. They've been incredibly successful in terms of giving us insights about at least the most common variants that exist across the world's populations. To give you a sense, when the Genome Project ended 15 years ago, there were probably maybe a few tens of thousands of places in the human genome we knew people differed. We now know it's approaching about 100 million places in the human genome that we know people differ, and we know what the different letters are at those positions, and all that's available to research scientists. And so we've gone from having very little information about human genomic variation to having very dense and rich catalogs that you could all utilize of the most common variations in the human genome across the world's populations. So that's wonderful. On the other hand, it doesn't necessarily, just because you have a variant, you can't immediately know whether that variant is functionally significant or not. Because what you need is knowledge about how the human genome actually works. And therefore, you'll know if a spelling difference influences the function of the genome at any particular place. How have we been doing in interpreting that human genome sequence? We've done well. We've a long way to go. We've done well. I think we have made profound advances in understanding how the human genome actually functions. But notice we don't have complete insights about how the human genome functions. So let me tell you what we know. Let me tell you what we don't know. So shown here is the human genome sequence. Well, actually not. It's only 0.0001% of the human genome sequence, but you get the idea. Let me be clear. The Human Genome Project delivered the sequence. It didn't deliver an interpretation. Never made that promise. That was what was going to happen after the Genome Project. It was going from this basic G's, A's, T's, and G, A, T's, and C's to actually knowing where are the functional sequences, where are the genes, where are the various other things. And that's a lot harder, actually, and will take a lot longer than the Genome Project. But we're making progress. Let me tell you what we know so far. We've gotten really good at understanding where are the parts of the human genome that directly code for proteins, in other words, protein-coding genes. And we have a pretty good catalog of all the sequences in the human genome that directly code for protein-coding genes, about 1.5% of all the letters of the human genome. A very, 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 very small fraction of the human genome directly codes for proteins. But we know we have about 20,000 genes. We know where they are. We can highlight them in yellow. We've got a great catalog. That's all wonderful. Here's the twist. The twist is that's actually not where we're complicated. We have a fairly, as a species, have a fairly modest number of genes, and our complexity is not in our gene number. Our complexity turns out to be what is often referred to as the dark side of the human genome. It's the side of the human genome that we actually know is functionally important because it's actually conserved throughout evolution. There's lots of places in our genome that don't encode for genes, and yet you can go back as far as reptiles and birds and find those same exact sequences that are highly conserved. Evolution has held on to them because they're incredibly important. We just don't totally know why they're important. So that's the dark side of the genome. And it turns out that we use a different color to highlight those. It's a much larger percent. It's upwards of probably 10 or 20% of our genome that's highly conserved, clearly functional, but is non-coding. And we're getting better at understanding this. These are sequences that are involved in regulating genes and playing other roles in how our DNA gets replicated and packaged and basically how chromosomes function. But it's still not quite apparent exactly how all those sequences work. That remains a pretty big challenge, although we've made great progress in cataloging these sequences and beginning to understand what they might be doing. The other thing we've learned in the last 15 years is that we can't just pay attention to the order of the G's, A's, T's, and C's in our genome, but rather there's yet a whole other genomic code. There's a whole other code that is related to epigenomics, where our DNA gets decorated. It gets modified with chemicals. It gets associated with proteins. And these epigenomic marks on our DNA are very important for genome function. But we've gotten really good at being able to detect those marks. And so there's this whole third language of DNA, or I guess here it's called the second genomic code, but it really is one where we yet need a third color to annotate what parts of the human genome get epigenomic marks and under what conditions and so forth. It's one of the ways the environment likely influences our genetics is through epigenomics, and we're understanding it more and more, but we have a long way to go. The other thing we've come to appreciate in the last 10 years in particular is do not think of DNA as a nice, innocent, linear molecule in the inside of cells, but rather you must appreciate that the genome is a three-dimensional structure. And we are learning that DNA in its sort of compact form in the nucleus of cells actually has some functional aspects to it related to different parts of different chromosomes that interact with one another in ways that we're just beginning to characterize. So there's a whole three-dimensional language that we're just beginning to appreciate, and we have a lot more to learn. So it is sort of, I would say, sort of a full-court press on trying to understand how the human genome works. We've gone from knowing about this much to maybe knowing about this much. Ultimately, we need to know this much, right? And so We have multiple ways we are continuing to approach this. We have major international consortium that continue to work together to try to do all sorts of computational analyses and laboratory-based studies to try to basically interpret the human genome on a large scale. There's a constant effort to go back and forth between human genome and model organism genomes and being able to use the power of model systems to interpret gene function and even non-coding portions of the genome to understand how those function as well. So a key interplay of understanding the human genome through the lens of model organisms. More and more and more, we need computational modeling. We need artificial intelligence. We need everything in data science that can be brought to this because that linear code is pretty complicated, and we need to basically try to figure out every little nuance associated with it. And importantly, we continue to develop new technologies like everything else to try to get us better and better insights about the function of the human genome. So we have a long way to go. I often refer to genome interpretation as a multi-generational problem. It won't just be my generation or even your generation that will fully interpret the human genome. I think it'll be your children, maybe your grandchildren's generation that will finally figure out what most of the functioning that's going on within those 3 billion letters. But we've come a long way. And having come a long way, what I would say is it's allowed us to now think about variation superimposed on top of genome function. So now we can know when there's a variant sitting on a functional sequence, it may influence the functioning of the genome. And that has allowed us to begin to understand which of the subset of genomic variants actually influence us and actually influence our traits, or in other words, our phenotypes. And we really have made progress then in starting to look at the traits we're really the most interested in, which is human disease. And again, while we have a long way to go, I would contend that here in 2018, we have made significant advances in unraveling the genomic basis of human disease. So let me tell you what we've learned, and I'll tell you what we still need to learn. To really appreciate human genetic disease, which basically all diseases have at least a genetic and genomic influence, you really have to appreciate that they come sort of in major categories. And the simplest, although imperfect way of thinking about it, are sort of rare diseases and common diseases. So let me tell you about each. So on the one hand, you have rare diseases. These are diseases that, like sickle cell anemia, cystic fibrosis, Huntington's disease. These are diseases that are rare in the population, but it turns out that they're sort of simple at a genomic level because it basically involves mutations that break a single gene. Most of these diseases are basically single gene or monogenic disorders, also known as Mendelian disorders after the famous geneticist Gregory Mendel. And you have mutations in the gene, you get the disease. There might be some other variants in the genome that might influence severity. There might even be some environmental contribution, but by and large, a broken gene, you get the disease. Here, there's been great progress because it's a simple problem once you have all these things like ability to sequence genomes and interpret genomes and so forth. And we've made great progress, especially in the last 15 years. Let me put a number on it. The day the Human Genome Project began, October 1, 1990, 61 rare diseases that we knew what gene was mutated to cause the disease. 61. Fast forward to today, that number is about 4,800, nearly 5,000. So that's remarkable. We now know the genomic basis of almost 5,000 genetic diseases, where once upon a time we only knew a few dozen. Now, there's still a couple thousand more that we haven't figured out. We're working on that. There's lots of major efforts internationally on that. But we still have made great inroads to that. Here's the thing. We should celebrate our success in understanding the genomic basis of rare diseases, but we need to also caution ourselves to recognize that rare diseases are not the major healthcare burdens worldwide. The major healthcare, it's not what fills hospitals and clinics around this world. What fills hospitals and clinics around this world are these diseases. These are common diseases. These are diseases like hypertension, diabetes, cardiovascular disease, mental illness, autism, Alzheimer's. These are diseases that are common in the population, but turns out they're complicated at a genomic level because it involves multiple genes. It's not a single gene that's broken that you get the disease, but rather it's multiple genomic variants that each have a small contribution to the risk for the disease. And as you can see from this pie chart, there's often and almost always a greater influence of other factors, environmental factors, lifestyle factors, and so forth. And so teasing out these small contributions end up being much more difficult. So where we've made great progress so far in rare diseases, and we can look at that in the rearview mirror, what I can tell you is for common diseases, there's only a little bit we can see in the rearview mirror. Most of our accomplishments are coming up down the road. We are getting better at teasing these out, but they involve very large studies, and so that you get enough statistical power to see these small contributions come up to be statistically significant. And so what you can expect, I can tell you, over the next 10 years is a better and better insight that will be gained about these small genetic contributions to diseases like schizophrenia and hypertension and diabetes and asthma and cardiovascular disease and so forth. And these projects are all ongoing. Practically every institute at NIH is working on them, but we're not quite there yet. But we're getting some early insights that are actually quite revealing and quite gratifying. So those are my first 5 highlights. I will tell you honestly that probably about 5 years ago, maybe 6 years ago, when I would give a talk like this is where I would say, do you have any questions? Because we're done. And the good news is I don't have to stop my talk here anymore because we actually get to add a highlight. I can now talk about vivid examples of genomic medicine that's now emerging. And that just wasn't the case 5 years ago. And it's very gratifying to see because it means we really are progressing down this journey towards genomic medicine. And I can really say for the first time, and I couldn't say it 5 years ago, we are beginning to bring genomic medicine into focus. We always knew it was out there somewhere. We didn't really know what it was going to look like. And what I'm going to describe for you now, actually, are just some of the earliest examples of where genomic medicine is becoming a reality. But believe me, it's the tip of the iceberg. And I've been delighted to know that there is a tip to this iceberg and that we could point to these things. It's actually interesting is that I think there's others out there even more optimistic than I am that genomic medicine has become a reality. I was really surprised 2 years ago when this magazine called Modern Healthcare did a survey of its readers. And they asked the readers over the last 40 years, what has been the top-rated set of advances? And they actually came back and said the top advance over the past 40 years in healthcare has been sequencing of the human genome. And I don't think it was because what we had accomplished in 2016. I think they saw this potential of what we were going to see sort of be influenced in medicine because of sequencing of the human genome. So what were these readers so excited about? And why am I so excited that I don't have to stop my talk at this point? Well, I think there's probably about 5 hot areas in genomic medicine. The first 4 in particular really are demonstrating that genomic medicine is here or beginning to be here in certain parts of medical practice. So I'm going to go through these fairly quickly, but I think they'll illustrate to you that it's not hypothetical anymore. It really is coming into being, and I think for those of you who are thinking about professions in medicine and heading off to medical school maybe in a handful of years, or some of you I know are even in medical school, this is stuff that you'll be a part of. Not just seen in the early day, you'll be a part of really operationalizing some of this. So what are some of these hot areas? Well, let's just start with probably the most obvious one, cancer. Actually, I've said nothing about cancer, but it is absolutely the hottest area in genomic medicine right now. If you're not aware of this, cancer is a disease of the genome. It is a genomic disease. A cancer is nothing more than a normal cells picking up mutations to the point that those cells no longer are in control when they grow out of control, and they form a tumor. And what we're able to do, by the way, is we can open up a tumor cell and read its genome just as easily as we can open up a normal cell and read its genome. And what's gone on? Well, among the many tens of thousands of human genomes that have been sequenced, many of those have been cancer genomes. Because around the world, cancer researchers have started realizing about 10 years ago that, wow, this would be incredibly valuable to start reading out the complete sequence of tumors and look at the most common genomic changes that take place in different types of cancer and different subtypes of cancer and subtypes, so forth. And it has been transformative. And I think all of cancer research and increasingly cancer care is going to change because of the infusion of genomics. I think about it in particular how this is beginning to, so it's giving us insights about the causes of cancer at a genomic level. I think about it from a diagnostic point of view, maybe again because I'm a diagnostician by clinical training. You know, for years, cancer histopathology has been the mainstay. A pathologist looks at the tumor from that patient and tries to figure out everything they can about the tumor in that patient. But all they can do is visualize what they can see under a microscope. But we're giving a new tool here because now we can sequence the genome of that exact tumor, and we can now make maps of the derangements of the genome in that patient, in that tumor. And we could augment basic information about morphology with information about changes in the genomic signature and use that information together to basically have much better ways of subtyping the different types of cancer, but also making predictions about prognosis and best treatment options. This is changing so many things about cancer, I can't even tell you. It's a talk in and of itself. But needless to say, for some types of cancer, mainstream to do genomic analyses already. I think that list will grow considerably in the coming decade. And by the time you guys are out of medical school, I guarantee you that genomics is going to be a mainstay of oncology practice around the world. Second area, hot because it's here and now. In some settings, I think this list will grow. It's a big word, but you can break it down. Pharmacogenomics, it's pharmacology and genomics. It's giving out prescription medicine based on genomic information. It is remarkable, and I think all of you are aware of this probably, how imprecise we are in the prescribing of medications. Many of you, or either yourself or your family members, have experience where you get a medication and either it doesn't work or it makes you sicker. And it's not because the doctor was mean and wanted to hurt you. It's because the doctor was blind to whether it was going to work in you or not, or your family member. You know, Nature several years ago had an article about this, and look at the imprecise nature of medicine. So shown here are 10 commonly prescribed medications where it is known that for every individual shown in blue where the medication works, or all the individuals in red where it is known that the medication doesn't work. The problem is you don't know when a patient's sitting in front of you whether they're blue or red. Except guess what? We're learning that increasingly the reason why some of us is blue and some of us is red for medication is because we have genomic variants in drug metabolism pathways that influence whether we're going to be a good responder or a bad responder. And so the idea is not designing the right drug for you, but selecting the right drug from you among a set of options. And so what pharmacogenomics is when you have to decide what medication to give a patient, pick a medication based on your unique genomic profile and knowing which variants influence which drug metabolism pathways, and pick the medication that's most likely to work for you. This was all hypothetical until we really started to get at knowledge about genomic variants that influence drug response. And now the idea is that it's here and now for some medications where you can take patients all with the same diagnosis, do a genomic analysis of them, and stratify them. Figure out the ones to give a medication to, decide the ones not to give a medication to, and so forth. And so while this maybe is sort of mainstream for a few dozen medications, it's expected that list will grow considerably in the coming years. Next hot area, absolutely here and now, and some of it here and now in this building itself at the NIH Clinical Center, the use of genomics for the diagnosis of rare genetic diseases. Now here, it has come fast and furious, and I've witnessed this, sort of amazed at how quickly we've gone from really not being able to use genomic information to diagnose a patient with an unknown disorder to now having this be fairly routine. For a long time they said, boy, wouldn't it be wonderful if we just sequence somebody's genome when they're sick and figure out what's wrong with them, especially children where you're seeing them for the first time. And that all seemed hypothetical until the year 2011. And then it was the definitive case, the first example, or first set of examples, this boy from Milwaukee and these twins in San Diego. Each of them were really heading towards a very bad medical outcome. And almost in desperation, a decision was made to sequence their genomes. And they were very fortunate that they could figure out what the mutation was that was causing their disease. And they were very fortunate, because not always the case, there was a decision that could be made about a treatment option based on the mutation that was found. And in both cases, that medical intervention worked. And these children are all alive. These kids in the bottom are in college and doing quite well. And it has been truly breathtaking to see this happen just in 2011. By the next year, Nature magazine predicted that sequencing was going to alter the clinical landscape. And then Nature predicted by 2014 that, you know what? If you don't know what's wrong with a patient after a standard test, just sequence their genome. It just makes total sense. And so what I can tell you is that around the United States, and actually in many countries around the world, literally every month, hundreds if not thousands of patients are getting their genome sequenced when they have rare conditions. They can't figure out what's wrong with them, sequence their genomes. You don't always get an answer, and even when you get an answer, maybe in about half the time, that's growing with time. And even when you get an answer, you don't always have a treatment option, but sometimes you do. And so it doesn't take very many cases where you actually learn something and actually can do something about it that says we should just be doing this. And so this is absolutely becoming mainstream. The other setting this is becoming mainstream and was first demonstrated in this hospital was this idea of what do you do with patients who have just never gotten a diagnosis. Sometimes have gone around a long time in their lives, in the case of adults, or many children, where they go from hospital to hospital, medical center to medical center, doctor to doctor, and they can't get a diagnosis. And the idea was, is there a way to use sort of a really critical final clinical evaluation coupled with genomic analysis? And shown here is the team of the Undiagnosed Diseases program that started here in the clinical center at NIH and actually was so successful at sort of demonstrating the value of having such an approach. And now we have now a national network of undiagnosed disease centers that basically refer patients in who don't have a diagnosis and use a combination of very rich clinical evaluation coupled with genomic analysis to try to get answers to these undiagnosed cases. And there's similar programs are popping up also around the world thanks to the kind of advances that were developed here. The fourth hot area is actually two stories. They both relate to pregnancy. And here, one of them is really hot. The first one, the second one is a bit of a question mark, but is worth all of you thinking about. So let me break down the genomics of pregnancy in a very simple way, prenatal, postnatal. There's two different stories. So let me tell you the prenatal story first. Well, the idea of prenatal genetic testing is not new. It's actually been around for decades. Many parents want to have genetic testing done of their children before they're born, mostly looking for abnormal numbers of chromosomes, such as 3 copies of chromosome 21 or trisomy 21. For a long time, this approach has been quite heavily utilized, but it's involved fairly invasive methods, such as amniocentesis, which I've been told is not so pleasant, and I'm also aware that it also risks the viability of the pregnancy at a very small percent. But it's been used for years to access fetal DNA. But you access fetal DNA, and then you could do various things, such as look at the chromosomes, such as shown here, a chromosomal abnormality of 3 copies of 21. But there's also been fancier methods shown on the bottom right to have readouts in a more sophisticated way. But it's still involved getting to that DNA in an invasive way. But guess what? You don't need to do it that way anymore, because these new methods for sequencing DNA have gotten so exquisitely sensitive that they allow you to analyze the fetal DNA that naturally sheds from the placenta and floats around in the maternal bloodstream. This is known as non-invasive prenatal genetic testing. It involves, as you can see on the upper right, a simple blood draw from a pregnant mom, and which pregnant moms get all the time. You can detect, using these fancy new methods for sequencing DNA, the same amount of screening information about abnormal numbers of chromosomes, and it is now heavily, heavily utilized instead of methods like amniocentesis. To give you a sense of how the uptake has been of this non-invasive prenatal testing, you can see that starting in 2012, worldwide, major uptick in the number of pregnant women that are using this test. And guess what? Nowadays, it's estimated that 4 to 6 million women worldwide are getting this non-invasive method. In fact, one could argue this is the #1 use of genomic medicine in terms of sheer number, 4 to 6 million tests performed worldwide. It's absolutely become standard of care. Amniocentesis has plummeted down, and this is sort of now taken over as the major tool used for prenatal genetic testing. That's prenatal, hot, very hot. What about postnatal? What do you do with a newborn? You know, Time magazine, for example, wondered if 4 years ago whether, by 2025 or so, everyone would just get their DNA mapped. I think they meant DNA sequenced at birth. But, there's a lot of questions associated with that. There's a lot of things we want to talk about or think about. Do we want to have a genome sequence of every child that's born in the United States? Who would get that information? How would we store it? Who would have access to it? What would you tell the child at what stage? How often would you interpret it? How often do you reinterpret it? Lots of questions. Unclear. We actually got together with a Child Health Institute here at NIH, and we now have a major program to study this as a research project. What do you learn from a genome sequence that you don't otherwise get? If you don't have a genome sequence, how valuable is that to pediatricians in early stages of a child's life, and so forth? And we're going to learn. There's a lot of logistical challenges, a lot of ethical challenges, and we've been studying this and thinking about this. And I think over the next 5-10 years, we'll get much better insights about the desirability of sequencing every child at birth. Some people strongly believe in this, and someday we might do this, but I think it's still the jury's out on whether we want to pursue that right now or not. There is one setting, though, where one of our research groups that we funded clearly illustrated a need. And that related not to healthy newborns, but to acutely ill newborns. And here we've seen the practice of medicine change very, very rapidly. What this investigator did was to basically focus on children who are acutely ill in neonatal intensive care units. And in most high-risk neonatal intensive care units, oftentimes the neonatologist can identify children where they simply don't know what's wrong with them. They don't know how to treat them. The best they could do is predict how many more days they'll live, and almost are giving up. And for those patients, what this investigator did was develop a way to basically do a very rapid genome sequence and interpretation, something like about 24 hours. And they are finding that in many cases, they can figure out what's wrong with the child, and in many cases, they can intervene and save the child's life. And so you don't need to have very many success stories in that setting to basically say this makes sense to do. And so a lot of neonatal intensive care units are now developing routines for very rapid genome sequencing to be able to use this as a frontline tool for those children where they simply don't know what's wrong with them, and those children are heading towards a bad outcome if they don't at least attempt to see if genomic information will aid their clinical care. Last hot area is not hot because we have accomplishment. It's hot because it's really challenging. It's something for many of you to think about as well. And it really relates to this idea that we need information systems to help us implement clinical genomics. Now, why is that such a burden? Why is it that we can't just sequence a patient's genome and immediately know what to do? Well, I'm very optimistic, as you can tell, and I'm very enthusiastic, but I'm also very realistic at times. And this is one of these areas where I have to be very honest with you in telling you what one of the barriers are, or what one of the barriers is. This is not a barrier. Generating a genome sequence nowadays it's actually almost trivial. It's not very expensive. It's not very hard to do. It goes on every single day on this campus. And genome sequencing is straightforward. That's not what's hard. What's hard is that we can generate a sequence of any patient here in this clinical center. The problem is when we have that sequence, that we can even run it through a computer program and spit out the 3 to 5 million variants that exist in that patient's genome. But, realistically, even when we go round on that patient the next morning, this is what we feel like. We look at that list of variants, and we actually have no idea what most of those variants mean. It's actually interesting. It's not that there's no knowledge about those variants, because actually, if you go into the scientific literature, every week there's another paper, probably dozens of papers that come out and say this variant means this, and this variant means this, and we think this variant might mean this, and this variant in this worm does this. The problem is a busy, rounding healthcare professional doesn't have time to cull through this huge literature and keep up with this incredible advancing knowledge about variation in genome function. So there's a gap in our ability to generate the data and our ability to interpret the data. And that gap grows every single week. And so the real problem when we're trying to hit this head on is how do we sort of make this reduced to practice so that a busy pharmacist, a busy physician, a busy nurse can actually use the information in genomics and immediately know what to tell the patient. Now, this should all integrate with the electronic health record. This should all sit there and be readily pull-upable on some iDevice or some tablet that you're going to use in a clinical workflow. But somehow we need to get much better at having a sort of a very simple flow of information so the busy frontline healthcare professional knows what to do when they encounter a patient with genomic information and what it means. We are not there yet. And so we're working on that. It's really a research project as much as anything else. It is a grand challenge. I would not be surprised if those of you who go into medicine and you start to encounter genomics, you will face some of this hurdle because it's not going to be fixed in the next 5 years, although we're working on it. It really represents a grand challenge, I think, for the next 5 to 10 to 20 years. So those are the hot areas. What I want to do, and I'm going to try to go quick, although I do want to cover a few more things, and there's plenty of time we'll still have for discussion, is just to really emphasize that in the 30-year arc that I've now described for you of genomics, and I've been there from the beginning, and it is remarkable how the field's relevance has changed. And I hope you can appreciate this, that when I got involved in genomics at the very beginning, it was only relevant to research. It was geeks like me at the bench or in the computer. It was just genomic scientists, just researchers. That was through the Genome Project. When the Genome Project ended, it did expand its relevance because we got healthcare professionals interested in this vision, this journey to genomic medicine. And they joined us in this marathon. But it was still just professionals thinking about genomics. But you know what? When genomics starts to touch cancer patients and their lives, and genomics starts to become relevant for the decisions about medications, and genomics becomes relevant to pregnant women and couples that are doing prenatal genetic testing, and genomics becomes relevant to families or friends, people who have rare diseases, all of a sudden genomics is not just about professionals anymore. Genomics is about patients. And once it's about patients, we all are patients or we are friends and relatives of patients. It's about society. So what's been very interesting is when I got involved in this field, it was just a field of scientists and then a field of medical professionals as well. But now, this is a field that very much is integrated in society. And over the years, we have really thought about the different societal challenges associated with genomics. Issues around privacy, now as genomics starts to touch genomics starts touching medicine, issues about literacy, our understanding of genomics, what our healthcare professionals understand, what our patients understand, issues about regulating genomics, issues about who pays for these genomic tests. So there are huge sets of genomic issues that all of a sudden become very relevant. I can tell you, my institute, once upon a time, just worried about the science. Now we got to think about a lot of these other issues, and it is an important responsibility we have in addition to seeing the realization of genomic medicine. So I just want you to really emphasize that, because oftentimes students will ask a lot of questions that are very relevant at a societal level. I don't want to lead you to the impression that we're ignoring these things. We're very much not only embracing understanding these things, we're actually studying them. And we actually have a major program to study the ethical, legal, and social implications of genomic advances, because we see how important these are going to be to understand as genomics increasingly gets infused into everyday life. But the last topic I wanted to cover really is about the future. And it's really the last journey that I want to tell you about, because I don't want to leave you with the impression that I think all aspects of human disease and all aspects of medicine are genomic, because it's not. It's not at all the case. I fully appreciate that. It's what I work on, but I realize it's bigger than that. And I want to share with you this early part of the journey of precision medicine, because I think for those of you, whether you go into research or medicine, you're going to see this become a reality, because it is in a very exciting, evolving area. What I will tell you is, when you look, and I showed you this pie chart earlier, I get very excited about the left side of the pie chart, because it's what I do. It's genomic variation. And the reason I can be really excited about it is because I'm riding a wave of technological advances that allow us to dissect the left half of this pie chart. But I completely understand and appreciate that the right side of the pie chart is incredibly important for human health and disease. But what's been lacking have been technologies for measuring health and environmental monitoring and lifestyle and so forth. But that won't be the case forever. And one could imagine a day where there'll be better technologies that will fill in very nicely the right side of the pie chart. This has led to this idea of precision medicine. So genomic medicine, just about genomics. Precision medicine, being more precise and individualizing the care of patients. And I think of precision medicine as just a more precise accounting of individual variability. I've spent over an hour now telling you about genomics and how genomics is one area of individual variability. But there's so much more to our lives, right? There's unique aspects of our physiology. We're going to get better at measuring that. There's aspects of our lifestyle that influence our health and disease. There's aspects of our environment. And then there's aspects of things that are sort of halfway between lifestyle and environment, like what we eat, what we expose our bodies to, and so forth. And so the question is, can we start to harness information about the variability in all of these different areas to basically come up with more precise ways to practice medicine? And this has become a major research area at NIH and actually around the world. And it really gets back to a fundamental question I asked earlier when I was introducing you to genomic medicine. It's really the same issue, just on a broader scale. You know, really, the way we practice medicine today is based on the response of the average patient. We will look back on how we practiced medicine in 2018 and think we were completely antiquated in how we were doing it, because it was just so, we were just doing it based on what a typical person did, not what based on an individual patient did. And we can do better. There'll be a tomorrow where medical care will be based on individual genomic, environmental, lifestyle differences that enable more precise ways to prevent and treat disease. I've highlighted the first of those three underlined words, genomic. And I think we're starting to do that, and I think genomic medicine is becoming a reality. But what's exciting is to think about how are we going to get more comprehensively from where we are today to where we're going to be tomorrow. And I've emphasized in my talk the genomics aspect of this, but there's more to be done. And what you're going to hear increasingly about is using information and big data information coming out in electronic health records. You know, when the Genome Project started, you know, there were almost no functional electronic health records in the United States, but now most sites in the United States have functioning electronic health records. Most of the information in any of your electronic health records just sits there and doesn't get analyzed for research, but that's changing. And increasingly we're recognizing if we can harness the information in our records, we can do a lot of data analysis that teaches us about individual variability. Oh, and could imagine a day where we have better technologies that will allow us to more accurately monitor our environment, our lifestyle, our physiology? Well, yeah, we can, because you're about to see those technologies come on the scene. And I think all of us recognize, and there's many articles that talk about all these incredible wearable devices and mobile health sensors that are coming on scene and are actually starting to be deployed in a clinical setting. recreational devices like Fitbits and the like. I mean, those are fun, and they teach us something about our physiology, but they're not clinical grade. Really good clinical grade things are coming, including the ability to analyze analytes in our blood and our sweat, and our ability to do much more robust monitoring of other aspects of our physiology. Could one imagine harnessing these technologies and using these in a way that measure in real time and collect lots of data about people, maybe over many years, and maybe on a very large scale? This has led to a number of population-based studies going on around the world. The one going on here in the United States that was launched earlier this year was something called the All of Us Research Program. And the All of Us Research Program is the U.S.'s major foray into the idea of developing a collection of individuals, a cohort of individuals who are going to be studied for many years, and we're going to basically learn a lot about precision medicine. This program is about recruiting about 1 million or more U.S. volunteers who are going to agree to share a lot of stuff about themselves: genomic data, lifestyle information, biological samples, and access to their electronic health records. This is going to be an incredible data resource. And what we're hoping is that people like you will actually analyze this data, because we're going to provide open, responsible data sharing, building in lots of privacy protections. But lots of people will analyze this data in the coming 10, 20, 30 years. And we hope we can learn a lot about all the small contributions to health and disease in the practice of medicine in a much more powerful way. And so making precision medicine a reality is really becoming a very high priority here in the US. There are similar programs going on in multiple other countries around the world, and a lot of coordination is going to take place among those different programs. But I would say that there's a lot of analogies, or many analogies, between the Human Genome Project that I saw 30 years ago coming on the scene, or 25 years ago, and this All of Us program here at NIH and spreading out across the United States that I see now. I'm very excited being involved in both of these. Actually feel very fortunate. And it is a sense of deja vu, because I cannot tell you that if you go back 25 years, the Genome Project was in about its 3rd year. Very exciting, very unclear how we were actually going to do it. But we could sort of see this made a lot of sense to be doing, and you could see a lot of benefits coming from the data that would be generated. It's exactly the way I feel about the All of Us Research Program. Still only about the 2nd or 3rd year, just standing it up, a lot of uncertainty, unclear many details. But wow, does it make sense to collect this data and have an incredible community of scientists being able to analyze that kind of scale of data for teasing out individual variability and its role in health and disease and thinking about better ways of practicing medicine. But let me emphasize a lot of uncertainties. We really don't precisely know the best way to implement precision medicine, no better than we know the best ways to actually sequence the human genome the very first time we did it during the Human Genome Project. So let me just leave you with a quote from this guy, because I actually believe in this. It's so much of, I think, what we do on these journeys, whether it be journeys of genomics or precision medicine. There's a quote from Einstein. It's a sign that I have above my door in my office. It says, where Einstein said, if we knew what we were doing, it wouldn't be called research. And I hope all of you doing research this summer realize that's what makes it so much fun. If you knew what you were doing, then you're just doing something that's been known. The exciting part about research is the uncertainty and the fact that you're out there doing something for the very first time and learning something that somebody else has never figured out. And that's the exciting aspects of this journey of genomics and precision medicine. So I will stop there. I will put in one more plug, which you can completely ignore, but if you're at all excited about what you heard about, I put out a monthly newsletter that my staff and I put together that give updates about genomics. If you want one extra email a month, you can subscribe to it. I won't be insulted if you don't. I just wanted to make you aware of it. And I'll leave this up if you want to write down that URL during any questions and answers. And I will stay here as long as any of you want to answer any of your questions. Thank you very much. And this is being webcast, so before all of you trip over each other, but just slowly come to microphones to ask the questions. And don't be shy, because I'm told that the NIH summer students are incredibly extroverted and will ask lots of questions. If not, I'll ask you questions. That's the flip side of it. Go for it. We're going to go to the person on the right, and then we'll go there. Okay. Go ahead.
All right. So you talked about the importance of data science in analyzing genomes and understanding what's to come. And then you also talked about lifestyle choices and looking at different omics like genomics and proteomics and even phonomics and physiology.
Yep.
So in your view, how would you see critical aspects of incorporating different types of omics in understanding basic physiology that relate to health?
Yeah, so there are probably a lot of different ways I could answer this. I mean, I wouldn't just limit it to omics. I mean, I wouldn't just limit it to genomics and proteomics. I mean, I would just sort of, I would probably cast it as there's going to be all different kinds of data that that will be generated or are available. I would throw in there electronic health record data, which isn't even research data, but it's incredibly valuable for research. Genomic data, as better technologies come for measuring proteins, it'll be proteomics, and then metabolomics, and then glycomics, and blah, blah, blah. And as Fitbits become embedded devices, we will have EKGs of people 724, 365. We'll have all sorts of, there'll just be immense amounts of data. I could just sort of stop at any one of these things and just think about how much better it is to have more data than a single clinical measurement of your blood pressure when you go see your physician. If you think about all that data and all these things, it becomes a data science challenge. I think it's the reason why I think the next speaker in this series is Patty Brennan, who's the director of the National Library of Medicine, and I'm sure she'll be talking about data science because it's a big part of what she and her institute, it's called the National Library of Medicine, responsible for. Underlying all of this is I think your generation will absolutely do scientific inquiry in a very different way than my generation did. I think I've seen some of this transition. Even when I had my lab, I saw the transition where all of a sudden everybody early on was working at the bench and pipetting. And then that was like the 1990s. And then by the beginning of 2000, they were all at the computer. And even the way we wanted to design labs here at NIH has changed. And I think there are going to be significant numbers of brilliant scientists in your generation that will never touch a pipettement, that I think will really be analyzing all of these data because generating the data is going to turn out to be very easy. And we're working really hard to make sure all this data is shareable so everybody can access it. So I just think that's the most, some of these incredibly exciting opportunities. It's just a very different way of doing research. It won't be for everybody. I don't mean to imply nobody will be pipetting, but I just think the center of gravity will be more on data analysis than data generation. I hope that helps. Yeah, over here.
So precision medicine is obviously very promising, but what is the role of precision medicine in reducing or eliminating health disparities? And how are we going to train the next generation of physician scientists to use that explosion of data to do just that?
Yeah, it's a great question. You asked the question about how do we deal with precision medicine. I would actually just broaden and just say, how do we deal with any of these advances, genomic advances, precision medicine advances, any of these? How do we, it's really nice. Go back to my genomics and society slide because, under that slide is just dozens of topics. How do we see these great, exciting advances benefit all? And wouldn't it be a tragedy if it's only to benefit the wealthy, only to benefit certain individuals from certain groups? We think about this a lot, and it's really no different than other aspects of medicine. And I mean, what I have found very interesting, even though I went to medical school, I hadn't fully appreciated until the last 8 years or 6 years that the second you start touching the medical ecosystem, you just completely don't realize how complicated things are. Because as a medical student or even as a resident, you sort of know that immediate environment. But oh my gosh, it gets so complicated, and there's so many. Medicine is delivered in so many different settings and so many different social and cultural contexts, and it's so easy to have a cookie cutter working in one place and not even realizing that it's a completely ineffective way to work in another place. So I don't have a simple answer because, as you know, health disparities is not new to genomics. It's pervasive. What I would say is the fact I think that we're talking about this a lot and we're studying this a lot, and I will tell you a lot of people at my institute are quite involved in thinking about what are the things we could be doing to get ahead of the problem. Because we will all be very disappointed if genomics exacerbates health disparities or precision medicine exacerbates health disparities. One of the things I will tell you, it's a detail, but it's worth stressing, the All of Us research program, as we go to collect these 1 million or more Americans, there is unbelievable attempts to recruit to that program groups that have traditionally not participated in biomedical research. And so you're going to see diversity, that the diversity of that cohort will be unlike anything that's NIH has ever done before to help think about the kind of issues that you just raised. It's a great question.
Thanks for the talk. My question is also about the All of Us initiative. And you've talked a little bit about making the data accessible to everyone and scientists around the world. And you talked a little bit about some of the issues that you face in trying to do that, privacy, HIPAA, all of that. I was wondering if you could just talk more about your vision in terms of what the access to that data will look like, what form that data will take, and kind of the future of that data analysis, basically.
Well, so, I mean, we have a lot of precedent to understand what access might look like, because all the genomic data we're generating now and all the data in many projects we're involved in, not my institute, many institutes, there are ways of doing this where you have to be sort of a credentialed investigator. You have to have your research institution support how you're going to deal with the data and what you're going to do with it and not do with it. So there's always we're trying to think a little broader than that. I mean, one of the visions that I know, I can just speak on behalf of the NIH director because he talks about this in public is that we know how to do that, but we also want to make sure we could think about that incredibly smart high school or maybe even junior high school kid, a citizen scientist who has some idea of what they want to analyze, and they really won't have a research institution behind them. We want to think about what's the way to sort of get them access to this, but in a fashion that we can make sure that we don't violate anybody's privacy and so forth. So there's a lot of very smart people working on this and trying to think very creatively. We want to really try to empower. We also think about, forget the high school stuff. I mean, in addition, think about, college classes and think about, computer science classes. Harnessing the energy of students in colleges in various disciplines, access to that data. There probably will be discoveries that will be made by projects coming out of some of these schools. And so we need to think about what's the way to get them access, but at the same time making sure we have enough protections that people's privacy isn't violated. So it's a big challenge, but it's being looked at already. It's being extensively looked at even before the first data starts rolling in. Yes?
One thing that you didn't touch upon exactly is why exactly is it important to be able to interpret the entire human genome? Like, I understand why it's important for precision medicine or to be able to identify one genetic disorder, but what's the benefit in understanding the entire thing? Like, obviously there's an intellectual benefit, but I'm just wondering what you had in mind.
So it's a great question. Here's what I would say is we don't know what we don't know. And one of the things that I will tell you is that there are some very strong clues to suggest that a lot of the genomic variants that play a role in common diseases are not in protein coding sequences. So there's I'm not going to go through the nitty-gritty detail, but it is becoming very, there's rare diseases, it seems that most of the time the mutation breaks a gene by breaking the coding sequences, has a mutation in coding sequence. It seems that for a lot of common diseases, we don't think it's necessarily the protein coding portion of the gene that is mutated, but rather it might be the non-coding sequences that regulate the gene. And that sort of makes sense, because one could imagine it may not be overtly breaking a gene so you have none of that gene product that causes disease, but rather maybe they don't make enough of it, or they make too much of it, or they make it at the wrong time, or they make it in the wrong place. And that gives a subtle little tweak to your system that gives you a little bit more risk to being hypertensive or diabetic. And so because of that, it's been greatly motivated to figure out what about the non-gene parts of the genome, because that may actually be incredibly important for understanding human disease. So that's a good reason in and of itself.
Thank you.
Okay.
Hi, thank you for the talk. It was very eye-opening. So my question is, you had that slide with all of the base pairs and the different colors highlighting with the purple. How do you account for the introns, exons, and epigenetic factors and compile that all into one sort of conclusion of treatment or diagnostic? What's your vision for that in the future?
Well, I'm not trying to vision. So one of the things you might, if you want to take a look, I would send you, do a Google search, you get back to a computer, to UC Santa Cruz Genome Browser, or UCSC Genome Browser, University of California, Santa Cruz Genome Browser. We fund them. And even right when the Genome Project ended, they created a visualization framework for visualizing the human genome. And it's just one of these classic browsers where you can zoom in and out, and you can get all the way down to a base pair. And what they've done is they've set up tracks that are horizontal. A sequence goes along top and then tracks. And you could ask any question you want. You could sort of say, okay, here I'm sitting in the middle of the hemoglobin, some gene, pick a gene, whatever gene you want. And you could zoom in and you can see right where the exon is. You can see where the introns are. You could see where are alternate splice forms, what are things that are conserved through different animals, where are small RNAs made. I mean, it just layers on data after data after data. So I guess that's my vision. It's a vision. It actually, you raise a question that, but then that's actually too complicated for a busy healthcare professional, so they don't really care what's conserved in chickens. They just want to know, what do I do with my patient? Right. And so then there's a whole other sets of ways of visualizing this for busy healthcare professionals, and that's the kind of thing we're working on. But at the end of the day, a simple vision is that most of our variants are not relevant for our health. And so we really want to get this down to the subset of variants that are relevant, and eventually you want to have something that it's like, if you encounter a patient and they're hypertensive and they have a variant here, don't give them this medicine, give them that medicine. I mean, but it's like a learning system that would pop up and tell you. And maybe that'll all get integrated with your electronic health records. That's sort of one vision.
Thank you.
Yeah.
Hi. I was wondering if you could talk a little about how you saw the Human Genome Project and genomics, kind of the lessons learned from that whole process informing the big data initiatives with EHR and things that are kind of more variable in how they're defined and collected.
So if I understand your question, but I'm going to say something, and then I'll answer, and then you tell me if I didn't quite answer it. I think you're getting at a very interesting point, is that the Genome Project in some ways was easy because we knew when we were done. It had a very finite goal. When you're done ordering 3 billion letters, you pop the cork on the champagne and say you're done. And we sort of did that in 2003, even though we didn't really have all 3 billion letters, but that's a whole other story. But we fundamentally, it was something that you knew when you were done. If I understood your question, there is sort of an issue around the precision medicine initiative. It's really, there's not a defined endpoint. It really is much more of we're going to create this playground, and people are going to learn a lot by analyzing that data. But and I think that's something we will face in the coming years. Of how do you know when to stop it? How do you know when to scale it up? What does it look like? And it's too early. It's, again, if we knew what we were doing, we wouldn't call it research. But I think you're making a fair point that the Genome Project was unique in that it had a very discrete finish line, a reasonably discrete finish line. And some of these other things I'm talking about aren't quite as discrete. Well, although there are odometer moments. I mean, the goal is enroll 1 million people. I know they had a party when they enrolled the first one. They'll probably have a party when they enroll the 100,000th. I'm sure there'll be a party when there's 1 millionth. But other than that, you're right. It'll be open-ended in some other ways. Is that what you were getting at?
Yeah, and just kind of how EHR data is so variable and.
So that's another good question. Yeah, I always want to temper my incredible enthusiasm with realism. Electronic health records, I don't know if people know much about electronic health records. They're a mess. I mean, they're different vendors, and the vendors fight with each other, and they're not interoperable, and it's really clunky, and blah, 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 blah. And I don't know anything about this because when I was a medical student, it was all, and a physician, it was all paper charts. So I just hear all these things. But they are a mess, and a lot of the data in it ain't so great. It's one of the by the way, it's one of the reasons why when you look at any of these studies that involve electronic health records, you always want to have big numbers, like 1 million or hundreds of thousands, because you got to be able to basically eliminate crappy data, and a lot of times a lot of the data in there is crappy. So you got to get by it by starting out big and then sifting out and throwing away. So, yeah.
Okay. So thank you for the lecture. Your enthusiasm is really infectious. So my question is kind of regarding the ethics of genomics in the future.
Yeah.
So I'm sure a number of people here have seen the movie Gattaca. So how can we ensure that in the future the genome isn't used as a status symbol or like a discriminatory tool?
How many of you saw the movie Gattaca? Oh, most of you did. I have to tell you a funny story, even though we'll run a little over time. People can leave if they have to leave, because I'll just keep talking. I have a funny story to tell you about Gattaca. True story. So true story is, so when Gattaca came out, it's been a while, right? I mean, Gattaca was a while ago. Yeah. When it came out, before it came out, Science Magazine was going to do a story about Gattaca, like a movie review of Gattaca. And so they called me up and they said, will you go to a sneak preview of the movie, and then we want to interview you afterwards. But we have to go tonight because they're showing it was downtown Bethesda. They had some theater they had rented out just for this sneak preview. And I looked at the calendar and I realized it was my wedding anniversary. And I said, well, I'm not sure I can go because it's my wedding anniversary. And the reporter said, oh, we could bring your wife. And so I said, well, we've been married long enough, maybe she'll think this is fun. And so I actually called home. I said, you want to go see a sneak preview of this movie? And she said, sure, we've been married long enough. That sounds more fun than it's. So we decided to go. So there I went to Gattaca, and my wife was on my right, and this reporter was on the left. And we got about 10 minutes into the movie, maybe a half hour into the movie, which is like one of the darkest movies you could ever possibly imagine. And I turned to the science reporter. I said, are you really going to write an article about this in Science? She said, no, I decided about 2 scenes ago. There's no way Science is going to want to write an article. So the article never appeared in Science, but I got to see the movie for free. But it's a true story. But so what do I think of it? On the one hand, when it came out, it was just ridiculous that they could sequence a genome that quick. And then I showed you the example of the kid in the neonatal intensive care unit. The movie is deliberately dramatic and raises lots of issues. But it does raise a lot of things to think about with respect to, is genetics everything, and genetic determinism, and how much of it, dare we find ourselves in a situation where we let genomic information somehow influence, what opportunities children have, et cetera, et cetera. So the one thing about it I would say is that it's not surprising that would get popular attention. It's one of the reasons why, literally, when the Genome Project started, our institute began our Ethical, Legal, Social Implications Research Project, otherwise known as the ELSI program, or not a project, a program, ELSI. We still dedicate 5% of all of our research dollars to studying the ethical, legal, social implications. Lots of issues around privacy, lots of issues around the use of genomic information. The challenges are always remaining. But, I mean, Gattaca I didn't think was a great movie, but I still think the issues are worth discussing. And another thing, I told the previous person to go do a Google search. I mean, you want to get a little nervous to emphasize your question. Do Google searches on genomic testing or genetic testing. I mean, there are absolutely, and I actually have meant to make some slides for this. I mean, there are absolutely companies that will test your children for what sport they should do. That just came out about a year ago. There absolutely are companies that claim they'll do genomic testing to tell you what cosmetic you should use, what person you should marry, what food you should eat. It just keeps going and going. And I mean, I don't think there's much scientific basis for most of those things. And so there's a lot of issues around making sure people understand what they should and should not believe. And the thing that worries me is if they hear enough about inappropriate use of genomic information and these recreational things that are total BS, I worry they'll lose faith in it when they become a cancer patient or when they have other applications where genomics like pharmacogenomics is going to be really important clinically. That's the concern. And obviously I don't want to see people be harmed because they actually believe some of this stuff. So, I guess Gattaca began to open our eyes to some of this. And then there's been, as many of you know, many companies like 23andMe, Ancestry.com that offer a lot of opportunity for people to access genomic information on their own. Okay? Yeah.
Hi. So in the slides, you've talked about how genomic medicine can take a really important role in treating common illnesses or illnesses with multiple genetic factors, but also you've talked about how the right side of that pie chart is also really important. So I'm wondering where that line is drawn. Specifically, what do you see as the final potential of genomic medicine in a vacuum, and how might genomic medicine of the future treat something like the common cold?
Yeah, so actually I might have misled you if your interpretation from what I said that when it came to common diseases that genomic information would actually necessarily be change how we practice medicine. I actually think that what we will learn over the next 10 years about the genomic variants related to common diseases will be very valuable for helping us understand the underlying basis of disease, may help us think about new treatments. I'm not sure I know how much it'll be helpful for changing the practice of medicine. And you almost hinted at that in your question. So for example, if I found out that somebody had 3 or 4 variants that may make them slightly more predisposed to hypertension, we also know that there's a bunch of other variants, and we also know there's a huge environmental component. I'm not sure that would necessarily change what they're doing. I think there's some big questions. Actually, I didn't mention 23andMe, but I alluded to it on the last question. That's one of the big questions around 23andMe-like information, is it gives you some insights about some of these common diseases, but there's so much more to the story. So I think some people are not so certain that genomic information will help make predictions about common diseases. On the other hand, where I think it might be helpful is if you have a patient and they're hypertensive, You might want to know among a set of known variants that play a role in hypertension, you might want to know which ones are they, and that may relate to the best medication to give them, because you might know that out of 5 pathways that can be deranged to lead to hypertension, this individual has pathways 1 and 3, and we know this medication influences pathways 1 and 3 and so forth. So it's a little bit not so much about predicting, maybe a little bit about stratifying and maybe making the best choice for medications. Does that help?
Yes, thank you.
Okay.
Hello. First of all, thank you.
Okay, we're going to take one more, and then I'll stick around, and people who still have questions come just see me in person.
First of all, thank you so much for the presentation.
Sure.
But I know that you're super, super optimistic about all these discoveries and advances in technology, but I'm interning in bioethics, and so the ethical part really concerns
rein
me.
In, aren't you?
No, but I want to ask you, I mean, do you feel like the bioethics, the ethics of this is holding back like the discovery or the advancement of technology? And on a similar note, how do you see policy like following, these advances?
So what I would say is, I'm not. I think if it's holding it back, I'm not sure I have any serious concerns about that. Here's what we do to, I think, more directly answer your questions. I didn't mention this earlier. A lot of our big genomic medicine research programs that we have, we always, or in most cases, are carving out bioethics research projects that are directly associated with them. And so what we're doing more and more, and this is going on at NIH and other places as well, is instead of saying, here's sort of these biomedical research projects, oh, and over here are some bioethics projects, and they'll sort of make each other aware of each other, we're doing much more integrated stuff. And I think there's some really good examples of what we've done in that area that sort of has helped the bioethics questions that are getting asked be incredibly relevant to the advances that are taking place. And so, and if nothing else, the data coming out of that helps inform policy decisions. And a lot of policy decisions that are going to have to be made are going to need research data behind them, because you can't make them in a vacuum. So, and I would also say that there's nothing exceptional about genomics, that if you go over the history of biomedicine, anytime there's advances, there's lots of ethical issues associated with it. Genomics raises a bunch of ones that are somewhat concerning to some people, and you just sort of deal with them one at a time, and hopefully you try to deal with facts and research data to inform those discussions. And at the end of the day, a lot of this becomes very personal. People can decide what they're comfortable with, what they're not comfortable with, and it doesn't have to be sweeping that everybody has to follow the same routine. So, well, thank you for your attention. Anybody has questions, I'll stick around. Happy to answer them in person.